This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 468, recorded on November 17th, 2017. This episode is brought to you by the Department of Microbiology at the Icahn School of Medicine at Mount Sinai. The department is presently looking to recruit prospective graduate students to apply to the program by the December 1st deadline. For more information, go to bit.ly/micromssm. This episode is brought to you by the Virology and Gene Therapy PhD program at the Mayo Clinic Graduate School of Biomedical Sciences in Rochester, Minnesota. The renowned Mayo Clinic Virology and Gene Therapy program is currently accepting applications until December 1st. If you want to be trained in the exciting fields of oncolytic viral therapy, gene therapy, vaccine creation, or basic virology, apply to the program. You'll hear more about those later. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me here in New York City, Dickson Despommier.
Hello, Vincent.
Nice day out.
It's gorgeous.
But winter is ensconced.
It is. It's on its way. You can feel it.
Although it's not officially here yet.
It's
right?
licking at your heels, basically.
Let's see what the temperature is right now.
I would guess 46 degrees.
No, I don't know what that. No Fahrenheit. 9 degrees Celsius. Is it sunny out or?
Yeah, it's very sunny.
Very nice.
Yeah.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It's 43 Fahrenheit here, 6 Celsius, and breezy. Got a ripping wind out of the northwest, about 20 knots.
Ripping wind.
Oh, yeah. Yeah.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody.
Hello.
I am now checking the weather for its official stuff, but it's kind of bluish gray, almost sunny outside. Looks like it's 40, 41 on a couple of apps, which means that in Celsius it's around 4 degrees, just like your refrigerator or cold room.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. We've got 82 Fahrenheit.
We don't want to hear this.
That's 28 Celsius. 59% humidity. That's a dew point of 66 degrees. So for people who live in drier climates, that might seem a little muggy, but it's not bad. And it's partly cloudy. It's actually basically a beautiful day.
Look at that.
And we have a guest today. She is right here with us, with Dickson and I. She is a associate research scientist here at Columbia University. Amy Rosenfeld, welcome to TWiV.
Hello.
And Amy is a virologist, right?
Technically by training, yes.
Technically by training.
I'm a virologist, but we diversified out.
You're a first-class virologist, I think.
Thank you.
And we're going to talk about the work you've been doing on Zika.
Yes.
But before we do that, we have a few items of business. First thing is, we're going to give away a book today, so stay tuned till later. It's a really cool book, and it's autographed by the author.
Really?
So do that. And then we have, Connor sent us something which is timely, so I thought we would bring this up.
Mm-hmm.
He writes,
Yes.
I'm getting in touch about the Tax Cuts and Job Act. That in its current form strikes 26 U.S. Code 117D. The purpose of 117D is to ensure that tuition waivers are not taxable income. Were the Tax Cuts and Jobs Act passed as is, this would open tuition waivers as taxable incomes that would ultimately need to be paid by graduate students. For example, a student on a stipend of $20,000 a year with a tuition waiver of $42,000 would see their effective tax rate become 31%, up from 7.5%. Gives a link for that. If you are able, please try to spread the word to your listeners that this is one of the most important times to talk to their congressional representatives in recent history. I've included several links to stories and resources that may be helpful for those interested in getting involved. I've been a fan of the show since 2014. You all have been a big part of me getting into science, which has been the best love of my life so far. I used to think that science was meant for someone a little more pedantic, but you all, particularly Dickson, showed me that passion is the important part. Anyways, as a student from a low-income background who is on his own in pursuing education, I'm feeling pretty helpless for the moment. But tomorrow I will be on the phone all day in between work and classes, and I hope you will feel compelled to join me. Thank you for your time and for all your good work. And he provides a number of links, and we have added links to various stories as well. Unfortunately, the House yesterday, or Thursday, right? Yes, yesterday, voted along party lines, although some Republicans voted against it by about 20 votes, I think, to pass this bill. It's now heading to the Senate, where I understand that tuition taxation is not part of the Senate bill, but it's still a terrible bill because it is effectively a massive movement of wealth from middle income to very, very wealthy people. It enriches the uber rich in this country. So it's not really a tax cut.
And screws the Uber drivers.
Screws the Uber driver. It's not really a tax cut. It's been sold as a tax cut.
It's a tax cut for 1% of Americans.
Yes.
It most definitely is, and for corporations
companies too. But this is a horrible, horrible bill. And let me just point out that golf courses remain tax exempt under the new bill. I wonder why. I wonder why. It's a horrible bill.
It's a mystery.
Everyone needs to call their senators. In the Senate, I think it's going to be close because I think already one Republican senator has defected, and you just need a couple more, and that's it. Two more. And they have so the House stuck on a thing to negate the obligatory Obamacare subscription,
That's
right?
correct.
Yes.
Which is going to gut Obamacare, and that, of course, was the entire point.
That was what they wanted to use to pay for the tax cut.
But it doesn't really.
Well, I understand that.
I mean, that's a pretext,
right.
And yeah,
I understand.
This does all the horrible things that people have accused the Republicans of doing, and it really does. And in fact,
goes a bit further.
The official assessment of it just came out today, and I think it turns out that just about everybody who earns less than $75,000 a year is getting a tax hike.
Right.
Even higher earning, like middle class,
and even higher, most of them are going to end up getting a tax hike until you get up into the rarefied 1%, where you start getting huge bonuses, which of course those people don't need. And it has just all kinds of really, really nasty stuff in it. And this particular aspect of the House bill, if that ends up in the final act, would of course be a complete disaster for science. And for most forms of graduate education, it's going to be a catastrophe.
Well, it's going to push the country way further back from other places scientifically because you can't recruit.
Right.
Because you won't be able to recruit. So why would I go to graduate school?
Not only can you not recruit, you're going to lose a lot of your current students because they simply will not be able to afford to continue going to graduate school if their meager stipend is now going to pay taxes, and they're going to have to quit.
Should point out that the real estate deduction is going to go, which would affect me. My taxes will go up. New Jersey, New York, and California have a lot of people using that. And of course, those are very democratic states. So that's really a screw you to those states. Really horrible bill that does not do anything for people. So I hope those of you out there take this into consideration, please, and call people and tell them not to vote for this because it's really bad. It's not for us. It is for the uber rich who will get more money, and they will be able to further influence the electorate, the elected officials. Did you know, for example, that the Koch brothers want to buy Time Incorporated now?
Yes. So now they're leaning on,
What a friggin' disaster that would be. Then that would go the way of Breitbart, all conservative lies.
Yes.
Oy. And Alan, can you take Sam's email, please?
Sure. Sam writes, Dear TWiV team, I've been listening to your podcast for some time now, and as someone without any formal education in virology, my MS is in chemistry, I found your podcast to be accessible and informative. I was especially interested in your discussion last week on the excess of scientists our current educational system is producing. I agree that this is a systemic problem that doesn't admit simple solutions, but I wanted to share my opinion on what may be at least a partial solution. By the way, with reference to the tuition tax, that would actually address this, but in a completely traumatic way.
Yeah.
Sam continues, partly because I saw the dismal career options at the end of a PhD, but mostly because I discovered that I don't love bench work. I revised my graduate education plans and decided to go to law school instead. Although my career plans are pointing me to patent law, I found to my surprise that there is a need for science-trained lawyers and judges in all areas of the law. Particularly judges, most of whom studied humanities before they went to law school, are routinely required to make judgments about scientific validity, usually when deciding whether to allow an expert to testify before a jury. If we had more judges with science backgrounds, we'd get more coherent decisions. As a graduate student, I got almost no guidance about how to prepare for any career besides academic scientist. Graduate students who expressed interest in other career paths were not viewed as being serious. If graduate schools started encouraging students to look seriously at so-called alternative science careers, law, journalism, politics, etc., it might kill two birds with one stone. We would have more lawmakers, journalists, and judges possessing basic science literacy, and we would reduce the imbalance between those seeking a PI position and the number of positions available. Anyway, those are just my thoughts. Keep up the excellent podcast. And yes, this is something that it was definitely true of graduate programs when I was coming through. The only career path that was seriously discussed was becoming a principal investigator, even though even at that time that was going to be a minority of us. And
Alan, I have to disagree because
really?
I think I went through before you, and biotech started to be discussed toward the end of my graduate career.
Which
so
was
there were
pretty
other,
much okay. There
were other options.
That's in my talk that I give on this. The industry shunt had been discovered,
Right.
but that's really kind of a parallel thing. It's not really a distinct career. I mean, the concept in going into biotech is that you'd be doing what a PI does, but you'd be doing it in a corporate setting.
I kind of disagree with that too, but
okay.
That's for another day. Okay.
All right.
The thing about law, I thought law was overpopulated, right? Too many lawyers.
Is.
And unless you're very conservative, you're not going to become a judge in the next 4 years.
That's true.
Right?
It depends. Well, right. And I mean, there's a disjunction between what one sees is needed in society and what society is actually going to do. So yes, I would agree that there is a need for scientifically literate judges, but what's actually going to happen is that those are primarily political appointees. Yeah. And, that's not necessarily going to be somebody with a science PhD and JD.
But again, if you don't start now having more people with some science background going to law school, then it's going to be a vicious cycle.
Yes,
so
and those people do have. I was just about to get to the point that those people have to exist for that to happen, and the politicians have to be willing to appoint them. Should there still be a United States 4 years from now, I think we may come around in a political cycle to appoint more people who are scientifically literate. In terms of the other career paths, I agree completely that these have not been discussed traditionally in graduate programs, but I increasingly hear from graduate programs who want somebody like me to come talk to them about pursuing science journalism, for example. So that is happening.
You came down to the University of Florida to talk about science journalism as part of our program to expose students to a variety of careers.
You need outside people because these PIs don't know anything except being a PI.
You're
right,
going
and that's
to Sinai.
What's that?
Aren't you going to Sinai to discuss that?
I'm talking about communication, not careers. That's different. I don't know anything about careers. I know a little bit about communication.
The biggest missing element, I think, in our society today, of all of the areas, is the elected officials. To see people in Washington that actually knows what the word science even means is essential. And we consider physicians as scientists because it's, we had Jimmy Carter. He was a nuclear engineer. He was a scientist, right? We had Michael Bloomberg as our mayor. And he got his degree in engineering from Johns Hopkins. And you can see the difference right away with those people because they understand the need for science and therefore they support it.
So why do you think so few scientists become elected officials?
I think because it's a dirty business to get there because you have to make friends with everybody and you have to pay them back when you get to the top, and the top meaning being elected. And therefore, I think that's why a guy like Bill Bradley just dropped out of the whole thing. He just had a belly full of it and said, I'm not going to do this anymore. This is not rewarding. He has a law degree.
So you're not exactly the proponent for the new HHS secretary who's a lawyer.
Oh, right.
Back when you were talking about the previous letter, I was on mute because I had sneezed. And I was trying to say that for people who might be listening and think it's too late to contact Congress, they're going on a week of Thanksgiving break. And so Senate's not going to take this up, as I understand it, until at least November 27th, if not later than that.
That's
so
right.
you do have time if you're listening right away to this podcast.
Definitely. Okay.
Will that be after the elections in Alabama?
When are the elections in Alabama?
December.
They're going to come after. That's right. That's too bad.
So, Amy, tell us a little bit about yourself. Where are you from originally?
I was born in St. Louis.
Louis.
Outside St. Louis.
Uh-huh.
And my father was a postdoc at WashU.
With whom?
With Roy Vagelos.
Hey, we know that name.
We do.
I'll be darned.
Yes.
He was a graduate of this medical school, right?
Was, yes.
And he's just given a lot of money
Yes.
to build a new building and so forth. Okay.
But at the time, he was a lipid chemist.
Mm-hmm.
And he had been part of the NIH. And then he was recruited to WashU to build the biochemistry department, which then knocked Urbana out of being the best biochemistry department at the time.
Ha.
And eventually...
How long did you spend in St. Louis?
I personally spent 2.5 years
Right.
there, but my parents spent 3.5.
Right. Where'd you go to high school?
In New Jersey. So we moved back when my father got a job at General Foods to decaffeinate coffee using bacteria. And I grew up for the remainder part of the time between the Borscht Belt and suburban New Jersey.
Mm-hmm. And I know you went to college at Emory, right?
I did go to Emory.
You went as a bio major?
Yes, I went as a bio major. And I had been interested in science for a really long time.
Gee, I wonder why. Now, your father was a scientist. What did your mom do?
My mother was a scientist also.
Mm-hmm.
They were a good pair. She did all the math. He did all the bench work.
Yeah.
And by the way, the Borscht Belt, for those who are outside the area, refers to the Catskills, right?
Indeed.
Yes.
Why is it called the Borscht Belt?
Because when you were living in the city and you were Jewish, you sent your family in the summer to the Catskill Mountains and live in these bungalow colonies. And then the husbands would come up on the weekend and participate in family events. And then they'd go back to the city.
Yeah.
That's interesting. I thought you were a bit young to have been part of that scene, but that was still going on in your generation.
Yeah. We went to the Nevele. The Nevele didn't close until like 1984.
Okay.
So tell us what borscht is.
Borscht is a cold Russian beet soup or Eastern European beet soup.
It's not just beet soup, though, right? It's got some other stuff in it.
Cabbage.
No, generally when you're an Ashkenazi Jew, you just make it with beets and then you stir in some sour cream or some
that's the missing. Yes. Or creme fraiche.
Yes.
So one of the things you do is you like to cook, right?
I went to cooking school for a little while in Montreal as a relaxing extravaganza from the lab. Yes.
So after college, what did you do?
After college, I thought I wanted to go to medical school. Then I didn't. So then I got a job at Sinai.
Mount Sinai.
As a technician in the department with Peter.
Yeah. And you work for Jim Wetmore, who I knew.
did work for Jim Wetmore. And then I decided after a year and a half, it's time to go to graduate school. I had ideas of my own and I wanted to investigate.
Mm-hmm. You came here.
I came here. It was the only school that was on the list that was approved. Wetmore went through the list and was like, nope. Oh, okay, we can apply here. It's like, okay.
They didn't apply to Sinai.
No, I did not apply to Sinai.
Of course, here you worked in my lab.
I did work and you were my third rotation, yes.
Okay. And then you did a postdoc, right?
I did a postdoc at McGill.
You might say it was plaques at first sight.
It was.
Flax. Yeah.
I worked on yeast, though. I didn't do virus.
Oh.
I did yeast in a virus lab.
She worked on yeast in my lab. Yeah.
He went through all of the list of all the projects, and I said, that's all very nice, but that's not what I'm going to do with you.
The only person that actually said, I don't want to do virus, I want to do this. So.
Oh, well.
And now you're back.
Now I'm back.
Now, when did you come back here? How many years ago?
About 3.
Okay. And when you came,
Yes.
There was no Zika, right?
There was no Zika.
No.
And then that happened while you were here.
Yes.
All right. And that's the paper we're going to talk about today. Your work on Zika.
Yes, my work on Zika.
Now, before we do, yes, did you have a question?
No.
Vincent?
I just want to hear ultimately more about the collaboration, how the relationships amongst the authors on this paper.
Well,
how this
we
came
will.
about.
We'll get into that for sure. I'll let Amy describe it because she's very fond of it.
I've heard a little bit about that too.
Yes, Alan has. I'm going to tell you about the sponsors of this episode first. This episode is brought to you by, look at that, Department of Microbiology at the Icahn School of Medicine, which is where Amy was a technician, right?
Yeah.
For 2 years?
Three.
Oh,
3.
three.
I was, 3
years.
3 years.
Well,
I wanted to finish.
They want you to know about their graduate and postdoctoral programs. This department is composed of over 20 virology labs, all in one building in the heart of New York City. Perfect fit for anyone with an interest in pursuing virus research. The department is presently looking to recruit prospective graduate students to apply to the program by the December 1st deadline. Interested postdocs are also encouraged to contact the faculty of interest. For more information about the department, go to bit.ly/micromsSM. Of course, many people that we know are at Mount Sinai. Let's see, besides Jim Wetmer, he's retired though, right?
Yes, but he was still there when I gave a
You
talk.
visited a few weeks ago.
Yeah.
So we have Peter Palese, we have Matt Evans, Adolfo Garcia Sastre, Ana Sesma Fernandez. Who else?
We have Ben.
What?
Ben Tanover.
Why are you so distant, Rich?
Oh, sorry. I'm messing around. Ben
You're messing
Tanover.
around.
Yeah, I'm messing around. I'm sitting back, Ben
Tanover,
Tanover.
right.
Did you mention Matt Evans?
Yeah.
Yes, you have Jane Lim.
Gene Lim,
Gene Lim.
who you knew from Florida, right?
Mm-hmm.
And they used to have Ann Moscona, but she's here now.
She's here now.
She is.
That's right. So, good program in New York City. And this episode is also brought to you by the Virology and Gene Therapy PhD program at the Mayo Clinic. They want you to apply to their fantastic program. The program has the stated mission to train students to work together with scientists and physicians to address important questions in fundamental virology, as well as more translational applications using viruses as tools for gene delivery or oncolysis. Students encounter small class sizes in courses taught by experts in the basic principles of virology and the translational applications of viruses. The Mayo Clinic grad program offers a competitive program with an average time to degree of 5.2 years, with over 90% of individuals successfully pursuing the career of their choice after graduation. The grad school offers a stipend of over $3,000 a year with special programs for additional financial assistance, especially for those from disadvantaged or underrepresented backgrounds. Through the unique funding mechanism of the Mayo Clinic Grad School of Biomedical Sciences, students receive their stipend from the graduate school, not from a PI. Therefore, the students are completely free agents, able to choose the lab and situation best for their careers, not based on extenuating financial factors. Students have no requirement to serve as TAs. However, opportunities in entrepreneurship, communications, and teaching are available through career development internships in partnership with local businesses, publications, and undergraduate institutions. Mayo Clinic is in the city of Rochester, Minnesota. It boasts a low cost of living, the perfect environment for a graduate education. Apply to this program, the Virology and Gene Therapy program, to begin your graduate education in the frontier of translational virology. If you want to be trained in the exciting fields of oncolytic viral therapy, gene therapy, vaccine creation, or basic virology, apply by December 1st. You can find the application at mayoclinic.org. Click on the Education tab, or just Google Virology and Gene Therapy and click on Mayo Clinic Virology and Gene Therapy for more details.
So these both have December 1st deadlines. Better get on it.
Get on it, yes.
And if you're applying to graduate programs, you can choose between Borscht and Mayo.
Oh. Oh, and if you're in southern New York, you can have that on rye.
Yeah.
Now, tell us how you started to work on Zika virus.
So we had started to work on EV68 or enterovirus T68 since you really can't work anymore on polio. And so I came in one morning and I said, we have all these tools. We should work on Zika because it's neurotropic also. And so you said, fine. You said, go get some virus. So we sat down and ordered virus and had it FedEx to us the next day.
Right. Now, if I remember,
and this was from the virus repository.
Yeah, at BEI.
UCC.
Yeah, you can just order it.
If you now, let me understand. So it's called BEI Resources, which distributes viruses for the CDC basically via ATCC,
Yes.
right? And now all we had to do was pay shipping. The viruses didn't cost anything. Is that?
We didn't even have to pay shipping at that time. That's only a new.
Yeah, that's a new thing. So why is it free? It's because it's a government.
It's a government thing, I guess.
Okay. They have other viruses as well, right?
Well, that's where we actually got a lot of the EV68 that we work on.
Right.
Also, we only collected one isolate from Ian Lipkin's lab. That was a clinical isolate, and then we have infectious clones of isolates from Europe. But everything else.
So, tell us how the collaboration with the Valley Laboratory started. I think you remember because I remember
Should
very.
we say who's on the paper first, so that they know who Valley is.
Yeah, we could do that. Go ahead. Well, do you have the paper in front of you there?
I know the authors by heart.
Do you know the title?
It's right there. It says replication of early and recent Zika virus isolates throughout mouse brain development.
Bingo.
And
this was in PNAS.
It was yes. It's actually the edition came out on Tuesday.
Yep.
And who are the authors?
So it's myself and David Dubin, who is an MD PhD graduate student in Richard Valley's lab in the cell and pathology department here at Columbia. And then for a bit of time, we had a second person in the lab with me, and that was Audrey Warren.
Technician, right?
Yes, Audrey was my technician.
And she had taken my virology course.
Yes,
And
the year before.
we responded to your ad, knowing it was in the lab of the person who taught the course, right?
Yes.
Okay. So how did
Vincent and Richard are also on the paper, of course.
Right. And so do you remember how Richard approached us? It was pretty interesting.
Yes. So he had a second graduate student in the lab who you were on her thesis committee. She did adenovirus trafficking along microtubules towards the nucleus. And she had heard through the grapevine that we had started to investigate and collect Zika virus isolates and started trying to understand how they cause neurotropic or neurovirulent diseases. And Richard works on microcephaly. And so he thought it was a perfect fit because he had known you had worked together for this graduate student.
Yeah, he emailed and said the grapevine. He used that word, the grapevine, also.
Yes.
Because I think maybe he should have been working on yeast.
Well, I feel like you need to have like a Marvin Gaye song, like
Yes. you heard about it.
Yeah, you needed to have the little raisinets with the legs dancing around.
Got it.
So he tell us. Do you know? Yeah, of course you do. What aspect of microcephaly does he work on? Obviously not Zika.
No. So he is a microtubule cell biologist, and microtubules and their motor proteins and many of the adapter proteins that link cargo to the tubules and the motor proteins are also critical for cell division. And he works on the part of microcephaly that results in aberrant asynchronous and asymmetrical microcephaly, because many children who have microcephaly have autosomal recessive mutations in genes that align the centrosome in the middle of the cell, and that's dependent upon the microtubules and the spindle. And so that's how he started many years ago working on microcephaly.
And tell us about the culture system that he uses to do that.
Yes, we've adapted their culture system. So he initially had a postdoc or a graduate student who came and had an idea that you can make embryonic organotypic brain slice cultures from rats because they're easier to dissect. The brains are easier to dissect out. And so that's how they study microcephaly, is they take embryonic rats at various time points during development, and they genetically manipulate the brain, and then they don't actually take the embryos out of the uterus. They do in utero surgeries, and then they sew up the mom, and then they come back several days later and they ask what happened to the brain in the presence of that altered protein.
So they introduce plasmids into the
They
brain?
introduce plasmids and small interfering RNAs to reduce the proteins. And then they also do, so then they, at the appropriate time points post surgery, they harvest the embryos, they isolate the brains, and then they either fix the brains and look just for gross anatomy defects, or they do live imaging on the slices to try to monitor what those manipulations actually do.
So wait a minute. I want to get this straight. Do they actually introduce plasmids or proteins into the brains of fetal rats?
Yes. But they don't introduce proteins. They introduce they introduce either nucleic acids, so it's either DNA or RNA. Yes.
Right, but right into the brain.
Yes. So you can take a very high power, I think they have I think it's a halogen lamp that they use. And when you open up the pregnant mom and you pull out the uterine horns with the presence of the light, the embryo, the uterine wall, and the skull are basically translucent. So you can directly see right into the skull where the ventricle of the brain is. And they use a very thin homemade needle to do this. Yes.
And then you just sew the mom up and she delivers normally? Is that
They don't wait for her to deliver. So you generally, well, I guess they and depends on the time course that they're doing, but generally when I'm there, it's like 96 hours, 48 hours after they've done this genetic manipulation. Yes.
Okay.
But sometimes I would assume that the mothers do deliver. Yes.
Okay.
So initially, he came with the idea of using rats, right?
Yes. Their system is all set up for rats. And we talked a lot about different microtubule proteins and various different things.
So the idea was to infect rats' brain slices and with Zika, right?
Yes.
So what made that not work?
So rats are kind of expensive, and they're
just bigger mice. Come on.
Well, yes, but mice are expensive.
And
they're more expensive than.
Tell me about it.
Time too.
Yeah, I mean, a mouse doesn't have a gallbladder; a rat does.
There you go.
But there's a difference between the cost of a rat and a heliotrope.
Yes, there is.
Which is huge.
Yeah.
There had been a lot of description of Zika by Dick in the 40s and 50s, and he had taken all these animals from the forest. Obviously, Zika was isolated in the Zika forest, and it looked for virus. And in not one of those papers did he talk about that he was able to find virus in a rat. So, being that rats are rather expensive, I thought we should look at rat cell lines first. And so we took rat 2 cell lines from Steve Goff's lab, and they came with, I think, a C6 rat cell line that they usually use. And I couldn't get virus to replicate efficiently in those cells.
Right.
And so that's why we stopped. We didn't pursue whether or not you can actually infect a rat slice.
But you decided to use mice instead,
Yes.
which they don't use in the Valley Lab. Is that correct?
They only use them when they're doing in vitro protein purifications from the brain.
Right. So you had to develop this slice system.
So David and I developed slices
from mice.
Yes.
Now, I remember when Valley came, he told us about how you can watch brain development occur in these slices. Can you?
Yeah.
Tell us how that works?
You can isolate after you genetically manipulate the brain as they do, and usually it entails electroporation of a plasmid that encodes for green fluorescent protein, or GFP. And by electroporation, you've injected the nucleic acid solution into the ventricular space and used electricity to facilitate its uptake to the cells that line the ventricular surface, which are the radioglial cells.
So this is cool. You actually shock the brain.
Yes.
To facilitate uptake of DNA.
It's 50 millivolts of current that go through like 5 times. You do 5 pulses.
And then you
So what he does, and because he's interested in the asymmetric division and asynchronous division of the microglial cells to establish to define the postmitotic neuron, they watch the green fluorescent protein producing cells move into the cortex and throughout the brain development.
So can you explain that? So there are stem cells lining the not the inside of the ventricle, but just above the ventricle, right?
Right. So the ventricle is surrounded by a basal membrane, and then these cells sit on the basal membrane.
And they're
I'm looking at this diagram that somebody put in the show notes.
Yeah.
That's really very helpful because I don't know any of this stuff, and there's going to be a lot of our listeners the same way. So the ventricle is the space in the middle of the brain, right?
Yes. So
and is that more or less acellular? What's in there?
So it's where the cerebral spinal fluid flows and flows out. So when the brain actually develops, it develops first you develop as neuroepithelial. So you make a tube that then you have to roll and close.
Mm-hmm.
And so when you roll and close a tube, you automatically get a lumen. And that lumen is referred to as the ventricle.
Okay. And
just to insert here, if listeners want to look at this figure, I don't know if Vincent will put it in the show notes for this episode, but it's also in his blog post from November 9th.
We'll put a link to both. Yep.
Okay, and then this ventricular zone, right? What you've been talking about, where you electroporate the GFP to basically tag cells from there and watch their migration?
Yes. So the brain develops in an inside-out pattern. So after you've defined the size of the brain, which is what the synchronous symmetrical divisions do, is they define the size of the brain. And then you're going to become a specialized cell. So you undergo an asynchronous asymmetric division. And a result of that asynchronous asymmetric division is the immature neuron. And that immature neuron migrates in an inside-out fashion. So you form the layers that are closest to the surface of the ventricle first, and then you form the outer layers last.
Okay.
And that migration, when Valley first visited, he said you can actually see the neurons moving up the fibers.
Yes.
Which I thought, wow.
You can, So we actually have been talking about making movies about labeled cells after viral infection. So you can put the slice, they have a CO2 chamber and a heated chamber on their scanning microscope, and you just set it, and you can actually make movies for days on end.
Watch the neurons. So you can label the neurons with GFP.
Yes.
By electroporating into the ventricle, it goes into those stem cells, and then they migrate up.
After they differentiate. So our idea was Audrey would help me make an infectious clone, and then we would electroporate the GFP encoding plasmid, come in with an RFP virus, and then watch the defect.
Now,
so the great thing about these slice systems is that, you've got the advantages and the manipulability of a cell culture system, but you will have all of the structure and the developmental program of the intact organ going on, right?
Yes. You also have other things. So since you're actually looking at the tissue, you have things that were not derived from that germline. So for instance, the microglial cells are present in an organotypic brain slice culture, but they're not present in an organoid culture, because they're not derived from the ectoderm. They're derived from the endoderm and migrate from the yolk sac into the brain at day 9. Or approximately
So in this case, you're looking at a slice of an actual brain as opposed to a synthetic sort of brain-like thing that you grew out of precursor cells.
So describe briefly how you make and culture these slices.
Oh, really? It's kind of gross.
Really?
So
I mean, it's the kind of stuff that I get into because I gotta conceptualize this, and I had to look up your methods and the supplementary methods to understand exactly how you do this. So you dissect out a brain. How big is a mouse brain?
depends on the age. It varies.
Oh, sorry, we're talking fetal here.
Yeah, fetal. Yeah, maybe a couple of millimeters.
Right, really small.
Yeah, they're not very large.
So this thing's like the head of a pin.
A little bit larger than the head of a pin, but yes. And the skull is very soft, but it's translucent. And so it takes a lot of technique to do this because you don't want to, when you're cutting through the skull, you don't want to cut through the brain. And then when you try to remove the brain from the skull, you use a probe and you don't want to cut off one hemisphere. Because if you cut off one hemisphere, that's not good either.
You according to the method, you embed this in low melting agarose?
Yeah. So you melt, like if you were pouring a slab DNA gel, it's basically low melting point agarose for a slab gel. If you were separating DNA bands of a certain molecular weight in artificial cerebral spinal fluid, and then you pour it into a Petri dish. And once it's pretty cool, like to your touch, you can introduce the brains into this agarose solution, and then you wait for it to solidify. And then as it solidifies, it fills in the holes and the creases of the brain into the brain. So it gives it some structure so you can get the blade or the vibratome through the tissue without fraying the tissue.
So then you slice it on a microtome? You freeze it and slice it?
No, you don't. No, because it's going to be live, so you can't freeze it. You slice it on a vibratome. So you put it in a chamber that is full of artificial cerebral spinal fluid, and then the vibrations with the blade help it go through the tissue.
I see.
But it's an actual blade that cuts.
Yeah, it's a razor blade, like if you were, shaving.
Yeah, and then you make pretty thick slices, right?
Yeah, you make them thick because if you make them too thin, then the tissue frays from the vibration.
So you take these slices and you put them immediately in culture medium, right?
You put them on a filter and you put them in culture medium, yeah.
And they're good for like a week and a half or something?
Yeah, pretty much.
And do you get more, you're talking about a fetal brain, so can you see significant development in culture, or does it stay put developmentally?
Well, you can see these neurons migrating, right?
Mm-hmm.
Yes.
Yeah, that's the main thing that happens.
Yeah,
neurons continue to proliferate and migrate.
Right. You can see, yeah. But if you isolate the brains from animals at different age points, yeah, you can definitely see the difference between an E15 brain and an E19 brain. And you can see the differences of like an E15 and a P1 or a P4. And the ventricle closes, and so as you age, the ventricle becomes smaller and
smaller.
Now,
if you take out an E15 brain and you've got it sliced and cultured, embryonic day 15, right, and you also take out an E19, embryonic day 19 brain, and slice it and culture it, if you cultured the 15-day brain for 4 more days, does it look like the 19-day brain, or do things start looking different in the slice culture?
Oh, good question.
No, it doesn't look like an E19 brain. It's in culture. It's going to start to die.
Okay.
It's not, you don't have all, it's not perfectly a perfect mimic of being bathed in the cerebral spinal fluid and all of the nutrients and all of the growth factors and all of the other stuff. It's not.
So it's mimicking many of the same processes, but this is not the exact developmental process, but it's as close as you can get and be able to get this level of manipulation.
Sounds like you can see a lot of the activities that were going on when you made the slice that will continue to go on, but not in a fashion that really results in a morphological change that you would see in development.
Well, I believe that you can actually do see that the cortical plate will expand as you age the brain.
So it doesn't totally arrest whatever development was going on, but it doesn't necessarily advance it to the same time point either.
Right. It's a hybrid. It's somewhere in the middle.
Which you'd kind of expect because you took a slice out of the thing. It's not the whole.
Yeah.
Yeah.
I mean, how does this help you study microcephaly?
So it helps you study microcephaly in various ways. So you can tease out the genetics very easily because you can generate the slices off of different genetic background mice. You can manipulate them and various other things. And you can also separate out the process of microcephaly. So microcephaly is really small head, and it's measured. It's a physical definition of a reduction of the circumference of a child's head in utero or after it's born. But now you can actually separate out whether or not you're talking about a proliferative defect, which is what we refer to as microcephaly, or you're referring to a migration defect, which gives you smooth brain, and that's lissencephaly, or if your gene also regulates both. So you get microlissencephaly.
So by proliferation, you mean proliferation of the stem cells to make postmitotic neurons, and migration is the movement up.
Right.
Right. And the thickness of the outer layer of neurons, that's a neocortex, right?
Yes, the cortex.
That is what is altered in microcephaly, right?
It's altered also in lissencephaly because you don't migrate properly. So like one of the first genes that's really well characterized is relin, which encodes for the relar protein. Relin.
And that's involved in microcephaly.
It's involved in it's actually a stop signal for migration. So it actually refers to the fact that you would get smooth brain, and instead of developing in an inside out fashion, you develop in an outside in fashion, so that the layers are all now mixed up instead of being.
Does Zika cause lissencephaly as well as microcephaly?
Probably. That's my hypothesis. Yes.
Okay,
but it's not been officially proven scientifically, but there is some indication from MRIs of children that they are microlissencephalic.
So now you took these slices and you infected them with Zika virus, and there's 2 phases of this paper. So let's talk about the first phase where you collected 15 different isolates. What was the idea there?
So the idea was it had been very heavily publicized that Zika had evolved neurovirulence and neurotropism. And I was trained by you. I can't accept that. It's not an acceptable rationale because I can't think of any virus outside of rabies that would escape the CNS. And so it's a dead end. And you have to continue to have new generations. So if you're in a dead end, how are you going to have new generations?
What benefit does the virus get from being neurotropic? Right.
So, Alan, you were trained with Vincent.
It sounds like it, doesn't it?
Like, you just can't accept these.
And by evolving, you mean from the original isolate in 47. We didn't see very little evidence of neurotropism until started to cause outbreaks, right?
Right. Yeah. But you also didn't see very many cases. You saw like 50 cases for 50 years.
Although there may have been more, we just didn't.
We didn't, right.
Yeah.
They didn't come in and say to the doctor, I have Zika virus infection today.
Okay.
So if there was neurotropism, there weren't enough data for a fairly rare event like that to show up.
Right.
And we should probably define our terms, right? Neurotropism and neurovirulence.
They're different.
Right. Tell us.
So neurotropism is, can you replicate within a neuron or a neural cell? And neurovirulence is, can you cause a neuronal disease? It's just like neuroinvasion means, can you enter the brain?
Right.
And they're all very different processes, but we seem to have mixed them up so that
we didn't mix. You and I don't mix.
We don't mix them up, but the community has mixed them up.
Can you just run through those 3 one more time? I think those are really important, and I think they bear repeating.
So neuroinvasion is moving from a site outside of the central nervous system, the periphery, into the central nervous system. So crossing the placenta, crossing the skin, crossing the bone, entering the brain, that's neuroinvasion. That's what 95% of all the work discusses. But they use the term neurovirulence, and that's not correct. What they're assaying is neuroinvasion. Neurovirulence is, can you cause a disease of the CNS?
And neurotropism
is, can you replicate and make productive infectious virus from a neuronal cell?
Right.
And you can only assay that.
Neuroinvasion, the virus comes through the door. Neurotropism, the virus starts raiding the refrigerator. Neurovirulence, the virus starts breaking up the furniture.
Yes.
Okay. And your slice culture system measures which one of the 3?
Measures neurotropism.
Right.
There's no animal to have a small head because the head is now not part of the brain, or the brain is not part of the head. And there is no placenta and there is no mom. So we're not talking about neuroinvasion.
And yet it has advantages over all other sorts of culture systems because you've maintained all of the cell types and organization that are in the brain,
right,
so that you can look at what happens on something that's very much like a brain.
So, Amy, how long does it remain essentially biologically viable for experiments?
Between 8 and 10 days, depending on the age.
8 and 20, that's a long time.
8 and 10 days.
That is a long time.
Yes. So if this culture system didn't replicate the brain, it would not have been well used to study electrophysiology of the brain. And that's what a lot of learning and memory systems are based upon, and Condit, Axel, Chessel's, a lot of their work all comes from organotypic brain size cultures and understanding the electrophysiology of the brain. That way, they don't actually stick the recording system into the mouse's head.
Or aplesia, depending on what
aplesia, yeah.
That's right.
All right, so neurotropism, you asked.
Yeah.
You collected a lot of isolates.
Yeah, so we collected isolates from 1947, the original MR766 strain from the monkey that was initially isolated, all the way through an isolate that we have from an outbreak in Honduras in 2016.
I mean, it's like 12 isolates or something?
I have 17, but there's an additional 3 that we've been discussing.
Okay.
Yes.
So you can just pipette the virus onto slices, right?
Yeah.
That's how you, and that's
you do a regular infection. You remove the media, you wash, so serum doesn't inhibit infection. You absorb the virus to the cells, and then you do that for a refined time. Then you wash away the inoculum, and then you add culture media.
And then you do the greatest assay of all time.
The plaque assay. Alan, you were supposed to chime in with me.
I was just coming in with it. Yeah.
As you've done lots of plaque assays, as I can attest to. In fact, we switched from 6 centimeter to 3.5 to save money, right?
Yes, I don't like them.
Yeah, so are you doing this in, you got the slices are really small. You do this in like a slice per well in a multi-well dish?
You do force, depending on the tissue. David and I did, depending on the age, I should have said, of the brain, we did between 6 and 4 slices per well. And you put the filter is adapted to sit in a 6-well plate.
Okay.
I wanted to ask why you do the plaque assay overlay now in 2 steps.
I've always done the plaque assay overlay in 2 steps with liquid overlay. Is that what you're referring to as the second step?
There's 1 step. Yeah, maybe that's what I missed. The first step.
The first step is the semi-solid agar.
Right.
And then I add a liquid overlay.
That has the other stuff in it, the pyruvate, glutamine, and oxaloacetic acid. Okay. Because when I got a protocol from Vincent, it was all together. So I just wondered if there's an advantage to doing it this way.
We never tried it separately.
No, we did try it.
It didn't work for you?
So it didn't make nice plaques.
So that plaque procedure was from Roland Ruckert originally for rhinoviruses. And Amy said, let's just try this. And it worked.
Yeah, well, first we tried
polio
plaque. Yeah. And I had adapted this one for EV68 and stuff. And so I had all the reagents sitting out because I probably hadn't fully stopped working on EV68. And I was just getting tired of having no plaques for like a week. And so I was like, okay, this is dumb. And we needed to evolve into a new plaque assay.
You should point out we've given this procedure to a lot of people, and some people say you don't need the second overlay, but we do. So it's obviously different.
It's just different in different labs. Different, maybe different incubators have different humidity levels or something.
But you use what cells to do the plaque assays?
You always plaque on Veros.
Veros, okay. African green monkey kidney cells, right? Very nice. I remember the first Zika plaque assay was very exciting. You know,
sure.
Dickson? Don't you remember?
I'm thrilled.
So I take it back.
Still thinking about it.
I pulled up the protocol that Vincent said, and it was also the 2-step protocol.
Because I when you email Vincent what happens is Vincent automatically forwards everybody's email to me.
Right.
And then I attach.
I don't send you my wife's emails.
Well, no, but
Remember, Vincent has the last say here. He can edit anything out of this.
but it's true.
Kathy, have you tried that protocol?
Yeah, my lab manager did do it. Yes.
Did it work?
Yep.
Okay, good. All right, so you did this, and you infected with all these isolates, and what did you find?
All of them replicated in the slice. We got infectious virus out after 5 to 7 days of culture.
So they're all neurotropic.
They're all neurotropic.
What cell? Do you know what cells they're growing in this slice?
We believe that they're growing in radioglial cells, the stem cell progenitors.
Okay.
You showed me that too. It was elegant work.
Thank you.
You're still working on that, right? Figuring out exactly what cell types.
Yeah, it changes as the brain ages. So as the brain ages, it no longer infects the adult stem cells of the brain. It moves into astrocytes. And that has been that was initially characterized in the 70s by the Dick lab also.
So if you the initial screen of all these isolates was done in E15 slices,
Yes.
right? And in E19, not all of them will replicate anymore.
Not all of them will replicate.
So just another point of interest here. How does the virus get from cell to cell in the brain?
So that is a debate that David and I have had for a while. So one idea is that you persist. So you see a lot of RNA persistence where they claim that there's replication because there's an increase by RT of RNA level by quantitative RT-PCR, but you actually don't get any virus out. So that would suggest that if you got infected and you persist, that as you divide and you migrate into different regions of the brain, that the virus is spreading through being like piggybacking into, because it's already there in the cell that divided off, so it migrates into the new region of the brain.
So it's not going from
it's not making an
neuron
infection,
to neuron.
right? It's not making an infectious particle.
Got it.
But there is some suggestion that it probably also disseminates by lysing cells and then going through the cerebral spinal fluid and all of the regular channels.
Now, in pregnant women, the greatest risk for having a child with microcephaly is if you're infected in the second trimester, right?
Well, that's when most of the cell proliferation occurs.
And that's about E15 in mice, right?
Yes.
And your isolates all pretty much infect, but E19 was a different story, right?
Right. So 2 of our isolates infected in an E19 brain.
And the others did not.
They did not.
So what do you think's going on?
Well, I don't know. I don't know if I want to say that they didn't infect. The infection did not result in the production of infectious virions.
You trained well.
Right. That's good job. So now it infects. So it's all these strains appear to be neurotrophic, and they also kill the cells, right?
They kill a significant percentage of the cells. Not all of the cells are lysed.
So while you can't necessarily conclude neurovirulence in the system because you're not getting a complete mouse out at the end, it seems neurovirulent in this system as well, right?
It seems partial neurovirulent.
Yeah, cell killing is part of that.
Yeah.
Yeah, sure.
Yeah.
So I think the not replicating in E19 or third trimester slices is really interesting.
Yeah, well, it also depends on how we're going to define replication. So what's
the production of infectious virus.
Yeah,
that's what I mean.
Thank you.
Yeah.
So there's some block, obviously.
See, he needs to be retrained.
I don't need to be retrained, but I think it would be interesting to figure out where the block is.
Yes, we're trying.
Because obviously something is happening between E15 and E19, and so if you could use the virus as a probe, that would be interesting to sort that out.
Yeah, well, at E19, you have the late stage of brain development, which is just gliosis, which is where you define the astrocytes and the oligodendrocytes. And you have a whole completely different transcriptional program than what you have when you're trying to define a neuron.
Yes.
So importantly, if I understand this correctly, of the 2 viruses that do replicate in the E19 brain, one of them is the original 1947 Uganda strain, and one is a more recent Honduras strain. So there's no correlation there between what replicates and what doesn't and the evolution of the virus, correct?
Probably, yes.
Yeah, the issue with the MR is that it was mouse passaged, right?
Yes.
So we don't know how that relates to the original.
Yeah, but
clinical isolate.
Having studied polio for so many years, one would never argue that passaging is a problem because that's how Sabin devised its strains, and we've used those attenuated strains to understand neurovirulence and neurotropism.
Yeah, but it could change the cell types that it's replicating.
But
the MR, MR and the Honduran in the E19s, they seem to infect similar cells, right?
We're
working on it.
Yeah,
Okay.
we're still working on it.
So there's a very narrow window that this virus actually has of causing disease then.
Well, in a mouse,
there's a big difference. There's a big difference between E15 and E19, right? That this all happens pretty fast.
Sure, but I mean, I'm thinking about all of the babies that are born for mothers who were positive for Zika and didn't develop pathology.
So that's very interesting because that was the initial assumption. But we now know that there are lots of mothers who are asymptomatic who could have microcephalic children.
But didn't.
No, they did.
No,
so they all have
a different question though.
Right. And there's also children who came from infected mothers who in utero looked to not be microcephalic, who then postnatally developed virus-induced microcephaly.
But then to Dickson's point, there are also a whole lot of mothers who were infected even during the critical second trimester who went on to deliver perfectly normal babies.
Right. So is there
and in fact, this has been a really weird aspect of the outbreaks, that there was this outbreak in northeastern Brazil where we saw this, and that seems to be strongly linked. And then in outbreaks before that people have gone back to, and outbreaks after that and elsewhere, you frequently don't see this link at all.
Well, that's not exactly true. So there was an outbreak of Zika in Hawaii, and then when they retrospectively went and asked, did we see an increase of microcephaly? They did see an increase that correlated at the time of the Zika outbreak.
I'm not saying the northeastern Brazil one was an entirely unique event, but it is perplexing that in other places that the virus has gone, like Colombia, like the Dominican Republic, you don't necessarily always see this spike of microcephaly along with Zika virus. There's not a clean correlation between the two.
There's not a clean correlation, but it's not as ambiguous as you're making it out.
Okay.
So
somewhere in the middle.
Right. Okay.
If the virus is neurotropic, then what does the mother get infected? What part of her body gets infected when she becomes infected?
So it's initially it was thought that we were only trafficking, or we're only looking at people who were infected by the virus through a mosquito bite.
Right.
And so you get infected through the skin. It traffics through the blood, forms of viremia, and then traffics through the placenta.
Wait, you said forms of viremia. Where does that occur?
In the blood. That's virus in the blood.
Oh, and what cells in the blood are they infecting?
So that's also a bit kind of unknown, but it's thought to be either macrophages or macrophages or some monocyte.
Really? So
or dendritic cells.
Have you tried any bone marrow cultures to see?
So actually, Daniel and I did CD34 positive cells.
Griffin.
Yes.
Yes.
So we actually did try to do Zika and CD34 positive cells, and it did not grow.
No, I meant a mixed culture from bone marrow that you just aspirate, put out as an explant, and then just see if you can infect any of those cells.
I haven't tried that.
Because
there are labs.
Is that a missing link in this story?
So Akiko's lab at Yale is looking at what cells in the blood are infected, and I think she's come up with dendritic cells. And she was here in June for the Parker lecture.
So cells that are infected must cross the placenta to infect the baby. Is that true?
I'm not sure. I don't think that's the working model.
Well,
the virus gets across.
Is that the virus gets across, not
free
the
viral
cells.
particles.
Free viral particles.
Yeah, I mean
I don't think all those cells can cross the four barriers of the placenta. But you can't really mimic placental transmission in a mouse and compare it to
No,
human.
it's got a different placental
It's
range.
totally different.
No, I totally agree. But
also, I mean, the other issue here is that the mouse gestation is 21 days, right?
Right. I mean, it's
and
quick.
humans
it's too quick. It's
months, right? 8 months. So
no, that's
it's very hard to pinpoint it down.
Oh, got it.
So then the other key part here is that you saw an effect of infection on neuronal migration, right?
Yeah.
Tell us about that.
So Richard Valley's lab looks at neuronal migration, or actually they look at the cytoskeletal architecture of the radioglial cells and how it's disrupted when they disrupt microtubules and their motor proteins. And you label microglial cells with vimentin because vimentin is part of the fiber that spans and is the signature mark of a radioglial cell. It spans from the ventricular surface to the peel surface. And so we labeled, using an anti-vimentin antibody, infected slices to see if the radioglial cells were intact. And the vimentin was no longer parallel. It forms a squiggle.
I think that's the most beautiful picture. Uninfected slices, you get these lovely parallel, very fine fibers. And those are the ones that the neurons are migrating on, right?
Yes.
And it's all squiggly. This is a scientific term, actually.
They're doing pull-ups.
After virus infection.
It is a scientific term. Arnold Crickstein writes in his reviews they form squiggles.
If you can have hedgehog, you can have...
So infection caused by Zika causes these fibers to squiggle so they can't bring the neurons up anymore.
Right.
And Crickstein has shown this in slices from human fetuses. Is that right?
It wasn't Crickstein. It was Brett Limbach's group at Yale in their cell. I think it's
They used human fetus.
They used human fetal organotypic brain slice cultures, and they saw a cytoskeletal architectural defect.
Do you know that this actually blocks movement of neurons?
It correlates with the impairment of the movement of neurons.
Because you counted them, right?
Yes. Well, David did all the,
Since some of the listeners might not have access to these figures, I want to point out that it's really nicely illustrated in Vincent's blog post, again, the one from November 9th.
Yeah, we picked two, the control and the infected for that, right?
Yes.
Mm-hmm.
There's one case where I could take a picture from the paper and put it in a blog post, which, by the way, was written by Amy and myself. There are two posts, one on the evolution and one on the effect on these fibers.
Yes.
So, Amy, how do you think the virus is making the fibers squiggly?
So we have two working hypotheses, and we're now trying to set up experiments to tease them out. So it could be that the virus directly causes the vimentin fibers to depolymerize or not be maintained properly.
Is there a cam associated with the migrating cells that it uses to attach to the fibers? Or how do you get it to...
Cell adhesion molecule.
Yeah. Wait, what was your question again? Sorry.
How do they actually crawl up the fiber?
They don't actually crawl up the vimentin. So the vimentin's internal, and then the neurons migrate on the external surface through NCAM.
NCAM.
So that's the NCAM.
So that's one of the molecules that
Okay.
N-cadherin is one of the molecules.
And what is the association there? Do you know?
Nobody knows.
Are there genetic defects of any animals or humans in particular and where the fibers are similarly affected? And you get learning defects and stuff like this as a result?
I think Richard's postdoc Orly has a paper on that.
Yep.
That talks about she sees squiggly vimentin fibers in one of her proteins.
It sounds important if it's critical for the development of the layers of the brain before you start to learn anything. If you're not set up for it, you're too bad.
Well, one of
right?
the yes. But it's been hard because one of the ideas that I had very early on in doing this work was that we should do the electrophysiology and the learn eventually.
Oh, yeah.
And it got poo-pooed as unimportant.
Well,
not by me.
No, not by you.
In parasitology, ideas you get poo-pooed get done.
That's great prediction. But this is TWiV.
The joke. All right, so obviously you want to know also what viral protein is causing the squigglies, right?
Right. So people keep writing in and saying it's NS2. And
what's that?
It's non-structural viral protein 2.
Mm-hmm. Does it have some activity?
No.
No, it's not. It's not a protease.
No, 3A is the protease,
Okay.
and 5A is the polymerase.
How many proteins altogether does it encode?
I think it's about 10 or 12, depending on the processing.
Okay.
And there are, I believe that they're not always fully processed. So some of them are like hybrids, the 2A, the NS223 protein. But they're not, it's kind of like a picorna where you get half processed proteins that have functions.
Yeah.
So, but all of that work that has described viral proteins in various aspects, like NS4A disrupting mTOR signaling, they're not ever done in the context of virus. They're just overexpressed proteins on a plasmid.
Sure.
And they never actually say, okay, well, if we make this mutation in this protein or in the gene that encodes for this protein and we have this altered protein, you lose the phenotype. So to me, okay, that could be a great guess, but it's just a guess.
So you want to find the protein that does.
So I just wrote a grant to do that.
Yes. Okay. I kind of know the answers to these questions.
Yes, I
You're
figured.
leading the witness.
I would hope you know the answers to these questions.
Why, Alan? Didn't you have secret projects when you were in Vincent's lab?
What kind of projects?
Secret projects.
Yeah, that's fine. Everyone should have secret projects. Yeah.
Not that I can disclose.
Exactly right.
Well, that's the whole point.
They did not, nor will they ever exist.
That's right.
You're taking the fifth.
Is there anything else?
It's less incriminating.
So anyone would like to know about this?
Well, we skipped over a part of this paper that I thought was actually quite interesting, and that is the apoptosis in the uninfected cells, right?
Yeah.
Go ahead.
She's cringing, folks.
He's cringing. She didn't like that part, right?
You didn't like that part.
No. So
Why not? Well, describe the phenomenon, then tell me why you didn't like it.
Okay. So what we observed so you either undergo division or you drop dead. And so you can drop dead in two different ways. You can either have a reduction in mitotic index where you're not going through the full cell cycle, or you can drop dead because you killed yourself by programmed cell death. And so we stained for cleave caspase 3 because that's the first step of the apoptotic pathway. And we saw that cleave caspase 3 signal was mostly in cells that were not co-labeled with an antiviral antigen. They surrounded the cells, and that occurred in like 91% of the time of the cells that were cleave caspase 3 positive. So my hypothesis was that the virus actually secreted something that was toxic to the cells, cytotoxic, and that it made a ring of death around the viral infected cell to protect the rest of the virus from viral dissemination. And
that's the message I got, and that's why I thought it was interesting. Yeah.
Yes. And that's because the brain doesn't really there's no innate immune response in a neonatal or embryonic brain. And this virus is extraordinarily sensitive to interferon in the innate immune response, because if you read all the papers, the only way it works in a mouse, you can get viral pathogenesis in a mouse, is if you use an immunocompromised mouse. So they either lack the interferon receptor or they lack the interferon alpha, beta, and gamma genes, so that they can't have an interferon response. One of the reasons why I don't like this is because lots of viruses cause apoptosis. It's not a Zika specific thing. I mean, hundreds of viruses cause cell death. It's not also clear to me that they're fully through the apoptotic pathway, that they're actually dead, because you're only assaying for one of the first two steps. And you eventually are an obligate intracellular parasite, so you need to have a live cell intact. So it doesn't really make very much sense to kill your cell or your host before you finish the life cycle, right?
Oh, it could be a host response.
It could be a host response,
yeah.
right? So my hypothesis is that it is a host response, and it's compensating for the lack that there is no innate immune response to restrict the virus otherwise. It's interesting in that context. I just felt that it was not very novel because, as I said, lots of viruses cause apoptosis. And so you're causing apoptosis in the brain. Yeah, well, I'm causing apoptosis in the brain because that's where I infect. I could cause apoptosis in the heart, but I'm not infecting the heart, so that I'm not going to see apoptosis in the heart. Some other virus could do that.
Well, one of the reasons that I liked it is that, in particular, in the context of this brain slice model or system, you can see that the cells that are undergoing apoptosis are surrounding infected cells and are themselves not infected. So it really accentuates this idea of the zone of death around the infected cell. So that's what I thought was kind of cool. If you do the same cleave caspase 3 staining on mock infected samples, do you see much reduced apoptosis, or is this something that's going on at a fair clip anyway?
No, you see a tremendous. I mean, I think it's supplemental panel 2 in the paper. You see
supplemental panel.
We had to. We couldn't fit it in the 6-page
kids
limit.
in these supplemental figures.
I hate them.
I don't even understand the whole point because it's online. Why do you even have a page limit?
Yeah, well, we were struggling because we could not fit this.
Yes, I know. I spent a day and a half.
Yeah, we were both struggling to get it. It's a 6-page limit, and the editor finally said, move some of the stuff to supplemental, and, I hate that, but we had to do it.
This is really striking.
What's interesting is that I'm an innocent bystander in this case also, and I could be knocked dead by something else, who knows. But I've been reading lately about the boycotting of Elsevier by authors like crazy because it's not an open source journal. It's a paywall journal, right?
Some of them are open
source.
It's
an enormous number of people.
The objection to Elsevier is not that it's a paywall journal. It's that it exhibits all of the worst characteristics of paywall journals that everybody in the open community has been railing against for years, and Elsevier has been further entrenching. And they've done some really repugnant stuff over the years, like the paid journals put out by pharma companies to promote dangerous drugs. But the thing that people are exactly, there's good money in it, but that shouldn't be the only standard. Yeah.
And I think they're now supporting NPR. Did I hear that?
Well, they've got enough money; they might as well.
I wanted to just address this one email that came from Emily, who's a PhD student at University of North Carolina, who asked us why we didn't publish this in an open access journal, because she says you're a strong proponent of publishing in open access journals.
Exactly.
Yeah, but she writes the question from her inconvenience, so this offends me.
I understand, but I want to address it from the point of why who I'm always talking about open access, did not publish this. So I would have been happy to publish this in PLOS Pathogens or MBio, which are open access. And I think you would have too, Amy, right? Or maybe I'm speaking for you. Would you have been happy to do that? Or
So I would have been happier if we were able to publish it in PNAS, taking advantage of their open access.
Paying for the open access.
The open access. But economics are extraordinarily tight.
Yeah.
And so
couldn't afford it. Yeah.
We chose to use that money to do experiments. I have a problem with the way jobs and grants are given out. And at this position and time, I'm looking for a job, a faculty position, and they just count papers. You've all just counted papers.
It's not just papers, though. It's where those papers are published, too.
And I've seen it. You guys just go and you count papers. This person has 3 cells.
Who are you talking to?
So general people who are on the faculty search committees, who are on the tenure track committees, people who are getting CVs from applicants, they come and they say, oh, how many papers does this person have and where did those papers go? Right. And when you look at the majority of people who have table of contents of journals automatically sent to them, they don't have MBio or JV or BBRC sent to them. They have Cell Science and Nature, PNAS, JEM, JCI.
Yeah, you're right. We've talked about that many times here.
We have.
I totally agree.
So at this point in time, it's not whether or not I believe in open access and whether or not you should have these, the community has given the power to these journals.
No, I totally understand. And I understand that you wanted to have a high profile.
Well, I'm having a difficult time enough getting a job, being as productive as I am. And they are only willing to look at me and consider me if I have these prestigious papers.
Yeah. Well, I was...
And you've all benefited, so unless...
Yeah, totally. I suggested other journals, but you and our collaborators didn't want to do it. We couldn't afford the open access at PNAS. So that's why, and I told Emily this. That's why it was. Although if it were just me, I would have put it in an open access journal. But
don't hate the players, hate the game.
We cannot it's sort of like Randy Sheckman, saying everything should be open access, where his career has benefited from being
tremendously.
Right, exactly.
He can do it at this point in his career, but we still have to solve this issue, and people still need to get jobs. I totally get that.
Right. And so you guys all actually had opportunities to change the rules of the game. And
I don't know about that.
Everybody has an opportunity.
The problem is that it requires action by everybody simultaneously.
Right. It requires a majority plus 50%.
It's like
plus
all the
one.
penguins standing on the edge of the iceberg, and none of them want to dive off first to where the seals and the sharks are, and somebody's got to do it. And it's just not happening. They're all just standing around doing the same thing. And there's a tremendous advantage to using this prestige-based system in that it's a whole lot easier. I mean, as a heuristic, it really narrows down your job applicant pool if you just say, well, how many cell science, Nature, PNAS papers do they have?
Yeah, but you're weeding out a lot of so
of
you're weeding
you
out,
are.
And you're not just weeding out a lot of candidates, but you're automatically making assumptions.
Yeah.
So
of course you are.
And but you at the end you have one phenotype, right? I either have a cell science or Nature paper,
right,
or I don't. And how I got there is not, you never know. So was I really not productive, or was I really productive, really smart, but just very unlucky? Yeah. And so
it's a terrible system. The problem is that the alternative to it requires a lot more work. And people are just so far, and I'm not saying, I mean, I don't sit on faculty search committees. I don't have to deal with this problem. But the alternatives would be to actually get into the nitty gritty of what did this person do and what is the real significance of it for however many hundreds of applicants you have for this faculty position. And I don't see that happening.
I mean, everyone has to simultaneously stop submitting to Cell Science and Nature, and that will never happen. The other thing Emily asks about, she says, I was wondering if you've seen this paper that describes the impact of editor-author relationships. It's called Quantifying the Effect of Editor-Author Relations on Manuscript Handling Times. It was published in Scientometrics. And she says, do you think having some kind of relationship with an editor gets papers into good journals? How should young female or people of color feel about this study, since they are generally disadvantaged by not having these connections? Do you have any thoughts about that, Amy?
My basic belief is if you deserve the journal, it'll eventually get into, if your work is good enough, it'll eventually get into the journal. And most of the time, it doesn't go by who the first author is. It goes by the senior author and what their reputation is.
So
it's not gender or color specific. It's about who the senior author is and what connections they have.
Mm-hmm.
It's not about gender or color, except to the extent that the connections of the senior authors tend to be about gender and color.
Well, and based on what's written in the abstract, I didn't look at the paper. There does seem to be evidence that it says even when correcting for other factors like time, experience, and performance, prior co-authorship relations have a large and significant influence on manuscript handling times, speeding up editorial decisions. So that's kind of the basis of this thing. So just whether co-authorship affects the manuscript handling time. But you could extend it or think about extending it to other parameters, like do the papers even get accepted, or even
reviewed,
things
right?
like that.
Right.
So I don't know why the time would matter if you knew someone, right? Because people say, I'm busy. I don't have time.
Well, time is money.
Yeah.
I mean, editor from a journal emails you and you say, yeah, I'm going to review that paper, how many of your reviews are actually submitted on time?
My reviews are all late, no matter who submitted the paper.
This is true.
Well, we know you're equal.
Equal opportunity for procrastinating.
Exactly. I just don't want to do it.
Yeah, we've all been down that road.
Yeah, right.
Does anyone else have any thoughts about this?
Just the same thoughts. That's all.
Rich Condit?
I was interested that the paper showed the correlation that Kathy suggested, that these relationships make a difference, at least in how fast a paper gets reviewed. It's no surprise to me.
Well, it doesn't account for how aggressive the person is. Like Vincent is not very aggressive, but like my postdoc mentor was very aggressive. He would call every day, maybe multiple times a day.
The journal, you mean, right?
Yeah. Saying, where's my review? And my friend who was a postdoc with, he would egg him on, or he would do it the call himself. Yeah. That's also might just be a reflection of the mentor, of the author. Like, you're not one who does that.
I mean, I certainly know a lot of the editors, but I still don't. Well, in fact, Cell Science and Nature, I don't know any of them because they're not scientists, right?
Right, but even still, like when I was a graduate student, we had a paper. You knew the editor who was handling it. He said X, and you just left it at X, and we went someplace else.
I remember Hillary Kaprowski called me once. I was handling a paper for him. He just called me and said, "Hey, Vince, where's my reviews?"
What are you talking about?
He didn't swear at you?
No, he didn't swear at me.
We just had that guy. There was a guy whose paper you handled for Plus Path or something, and it wasn't reviewed in a timely fashion. I think he called you like 3 or 4 times over the week.
Well, calling is not useful for me because I don't know what a phone is, right?
This is all
email.
Whatever.
Well, I if it's a problem and the paper suggests that it is, then it's just like the other publishing problem we've been talking about. I don't know what the solution is.
I mean, it's getting a job. It's like any other field. You make a deal. Somebody makes a deal in banking because they know the person.
No, I hate that. I hate deal making.
Remember my joke on this.
What's that?
When the Harvard graduating class was addressed by the president, he said, "Now remember, class, it's not who you know that's important.
It's whom."
Thank you.
Okay. All right, let's do some picks. How about that?
Picks.
That can be a little more uplifting.
Here we go.
Your paper was very uplifting, Amy. The subsequent
extremely
discussion on publication I find disturbing, but picks are always uplifting.
They are.
Now, let's start with you, Amy. Do you have a pick? What do you have?
So there was an article on CNN that talked about the forgotten Zika babies, especially.
This may not be an uplifting pick.
Yeah. It's okay. You can go ahead.
Well, it's not an uplifting pick.
It is a good pick.
In that respect, but I think it identifies a huge problem, and that microcephaly is a long-term disease, and we're just starting to understand what Zika can do. And it gives it, I mean, from a research perspective, it's kind of uplifting because it gives you like a whole new avenue to investigate and all these questions that you didn't even know about. And so there were 2 women who went to Brazil, and they wrote about all the some of the people and patients and problems that they encountered. And then they have this Facebook page where you can track them and make donations and various other things. And so I think it's fitting with the paper.
Cool.
Well, and I thought what struck me is that they talked about at least one of the mothers whose baby didn't really have the outward signs at first. And so they sent her home and never called her back. And so the baby isn't really getting any appropriate
so that's
treatment.
What I was saying earlier is that it's, yes, the first initial phase and the first huge observations of the disease were microcephaly, second trimester infection. But that dogma has to change. Like, you can no longer just say that that's true. You can get postnatal microcephaly where you were not microcephalic at birth, and now you have these huge defects as you mature throughout the postnatal developmental program. And so I think that is an example of where, science has, the research community has kind of said, okay, this is the only topic we're interested in, but yet there's all this other stuff. And
I also want to point out that before Zika, there was microcephaly.
Right.
Nobody pays any attention to that. Right.
But it's just like after polio, there's acute flaccid paralysis, right?
Yes.
And it's on the rise. And we can discuss that a different day.
No, but I agree that Zika exacerbated the problem, but there was before, and those people have a lot of problems as well.
Exactly.
This is genetic microcephaly.
And
there's even viral.
You get CMV induced, and there was an article about that too, about how when you go to the OBGYN, they don't even tell you anything about the fact that you can have a CMV infection.
Yeah, I mean, rubella still causes microcephaly for people who are not vaccinated.
Right.
So, I talked to reporters about that when they were talking about Zika. They had no idea that other viruses caused microcephaly.
Yeah, I mean, I have a cousin who had encephalitis because her mother when she was pregnant with her, had German measles.
Thank you. Alan, what do you have?
I have some cool animations, science education animations. The one I've linked to is just happens to be the first one I saw on this particular YouTube channel. These are clay animations about entomology. They're produced by the Cornell Entomology Department. And this one, the one I've linked to, is about the soil food web. Just, these interactions between various organisms that are probably in your backyard and you've never noticed them before. And there are a whole bunch of others. There are ecosystem services in agriculture and life cycle of the squash bee. And I just find these really fun. And I think they're just a really good approach to a topic like this.
Great. And they're short.
They're short, yes.
And because they're cute, they're engaging too. This is cool, Dickson.
Yeah.
Food webs, Dickson.
Hey, there you go.
This is your shtick.
It's my shtick.
That's really cool.
It's everybody's shtick.
Rich, what do you have?
I have 2.5 picks.
Wow, that's a record.
The first 1.5 were actually we didn't do picks last week because we ran out of time, but they were relevant to John Udell's presence. One is called "Letters to a Young Scientist" by E.O. Wilson, famous ecologist. Advanced in his career, years-wise and otherwise. And it's basically advice to people considering a career in science coming from another perspective. And I thought people might be interested in that.
Can I read it too? Is it relevant for me?
Sure, why not? You'll like it. The half pick is a TED Talk. Look at the TED Talk first. Decide whether you want to read the book. The TED Talk he wrote, it's basically based on the book, and it's as he was writing a book, he gave the TED Talk. So the other unrelated pick that I just wanted to get out before it gets stale is a Washington Post article called "Chasing a Killer" that describes a CDC investigation of a monkeypox outbreak in the Congo. And it's just very well written. And actually, I like the way they use the internet in this article. There's a lot of interactive stuff if you actually look at the Washington Post article itself, and it's just very nicely done.
Mm-hmm.
Cool,
Right.
Good example of science writing.
Richard, I once had the privilege of introducing Edward O. Wilson here.
Oh, cool!
Yep.
Excellent.
Yeah, we picked him for our environmental health sciences lecture of the year, and he gave a wonderful talk.
Well, he's an interesting individual.
Yeah, when he signs his books, and I had lots of them in my office. One of them is called Ants, because he made his fortune studying ants.
His fortune.
Yeah, that's right.
He made his fortune writing books.
And when he actually signs the book, he draws a little picture of an ant.
Yeah, that's cool.
Then he puts it next to his name. I'll try to scan that and send it out to you.
Should I write a little icosahedral capsid next?
You should. Yeah. You should.
I'm not very good at that.
Well, you have to practice.
Practice does make.
That's how you get to Carnegie Hall, by the way.
I got real good at icosahedrons in organic chemistry.
Dickson, do you have a pick?
I do. I just saw this the other night. This is not from Cosmos magazine, by the way. This is from a television series called SciTech Now, and it's a PBS show run by Harry Srinivasan. And one of the segments, the last segment of this last show that I just saw, challenged graduate students to convey to anybody who will listen in 2 minutes what they're doing in the laboratory, because the laboratory is kind of like a secret place that only scientists go, and they speak another language when they go inside the doors. But you can't do that if you're going to communicate your ideas to, like, say, Congress, for instance, and to the public, to make sure that they understand what science is all about. And this is a wonderful way to introduce that topic to graduate students, I thought. So if you watch this video, they offered a $750 prize. The graduate students lined up as though they were going to give poster session lectures, and each one only had 2 minutes. And a judge came by, and they had a bunch of categories in which they were being judged. And obviously the person judging this presentation had no prior knowledge to what they were going to say. And the winner was absolutely spot on in terms of his delivery. But a lot of other people tried very hard also to put into common language the essence of what they were doing in the laboratory. And I think we've talked about this a lot, but here's a mechanism for instituting this as an exercise at college level. I mean, this was done at Penn State University, and I think that this could be done in a lot of other places also.
Yeah, we violate that here on TWiV.
Well, we certainly don't take 2 minutes, do we?
Well, the two things, the internet goes to extremes. The two things that do well online are really, really short takes, like tweets.
Yeah.
Or 10,000 word long reads.
Yeah, no, you're right.
It's the middle ground that's a problem. So I think we're at the 10,000 word long read stage.
Yep.
Speaking of tweets, has everyone noticed that it's now 280 characters?
Yes.
That's right.
I kind of like that, because I always used to run out. I don't need the whole 280, but I often need like a few more extras. And it's really nice.
So you make them 2 tweets.
However, for a certain individual, it's not good to have 280 characters.
Yeah, right.
But I think that since they doubled it, they ought to call them twoots now.
Twoots?
Yes.
Kathy, what do you have?
I have a pic that's from the U.S. Geological Survey. It's a map called A Tapestry of Time and Terrain. And it shows not only the typical 3D perspective drawing of landscapes that, you can see where the mountains and the valleys and things are, but it adds into it the ages of the rock underneath the surface.
This is very cool.
In
I love this.
About almost 30 colors, I think. And so, just for example, what caught my eye is this blob in the middle of Texas that I've kind of triangulated is kind of northwest of Austin, about halfway between Austin and I-20, between Austin and Abilene. And it's way out of time with the surrounding stuff.
There's another similar blob north of that also, I notice. Up in Kansas, maybe.
So what is the oldest and the youngest? What's the color range? I can't tell from this.
So the old is toward the red, right?
Red, okay.
Yeah, the Appalachians should be the reddest, actually.
Yeah.
So what's on this webpage isn't that high resolution, but you can download the 45 megabyte version, a PDF.
It's fabulous.
Check that out.
It's cool.
Anyway, it's just,
I love it.
I really like it.
I never see this kind of information on a map, right?
No. And,
because the river valleys are pretty neat. The Mississippi River.
They are.
Well, the Mississippi is the main one. Right? Then you got a little coastal thing happening in Florida there in the southeast.
Yes.
And then a big one in the middle, too. That's very cool.
Very, very
Kotke.
nice.
I know Jason Kotke. Well, I know of him. Didn't know he did this.
Mm-hmm.
Love it.
All right, so I've been reading a lot of Nick Lane lately. I saw him talk at ASM in New Orleans this summer. He gave a fabulous talk about the origins of life. And I read his first book, The Vital Question. I think that's his most recent one. And this was my pick on TWiV 21. That's all about the origins of life and how he uses this idea that, all life is driven by proton gradients. And so he looks for the place on Earth where that could have arisen. And these are these alkaline vents on the bottom of the oceans where you could have a proton gradient. Really cool stuff. And he supports it with evidence and just, he's a scientist, but he's pretty accessible. Now, the one I'm reading now is called Oxygen. Oh, man, it's so interesting. Oxygen, where did it come from? How did it increase on Earth? How did it give rise to explosions and species and so forth? Why don't other planets have it?
Is this a religious treatise?
No, it's not religious at all.
Priestly?
He talks a lot about Priestley.
Of course.
One of the early oxygen scientists, Lavoisier, he was beheaded.
Yeah, of course.
And someone said, it took 10 seconds to cut that head off, and it will take us 100 years to get another one as good as that.
That's a great quote. That's a great quote.
That's what they were saying. But, oxygen is so
do you know
cool.
what happened to Lavoisier's wife?
No.
She was in France. She fled to England and married Robert Hooke.
Oh, yeah?
Yeah.
Really? That's
wow.
Cool. So this is oxygen, the molecule that made the world. He talks about, its origins, little history, where it came from in the beginning, what we thought happened, and what we know happened, and how you can tell by different kinds of surveys when oxygen levels change. It's just so interesting.
Yeah,
it's so
cool.
interesting. Early in Earth, the Earth was bombarded by UV, and the oceans, and the water was broken down into oxygen and hydrogen.
Right.
And that probably happened on other planets like Mars, but all the hydrogen leaves. Okay, the oxygen reacts with iron and becomes oxides.
Right. That's right.
And then if all the hydrogen leaves, you have any more water left, then you're screwed. But Correct. He said on Earth, what happened is that probably life arose and started pumping oxygen to by photosynthesis and so forth.
Well,
and
we got hit by comets too.
That kept the oxygen.
I think
for the first 500 million years, the Earth was bombarded by asteroids continuously. Yeah. Oh, this is so cool. Anyway, so Kathy, that's you got both links. Vital question. I will start with oxygen. I'm reading now. He's got two more. One is called Life Ascending, the 10 greatest inventions of evolution, and then he has Power, Sex, and Suicide, Mitochondria, and the Meaning of Life. And then he's into mitochondria. He talks about that a lot in the Vital Question, and how apoptosis evolved to make sure the mitochondria were functional, because if they started leaking electrons, that would trigger apoptosis. It's not viruses that did that. It's actually making sure the mitochondrial and nuclear genomes were in sync with each other, because the proteins in the mitochondrial membrane come from both. It's just so cool. And love it. Just love it. Highly recommend it. Oxygen and the Vital Question. We have a listener pick from Islam Hussein. Downcast.app.com, a podcast that syncs across all Mac and iOS machines. I bought it a few days ago. Have been enjoying listening to TWiV and all other podcasts on it. You can subscribe, create playlists, and they will sync across your Mac, iPhone, and iPad. I love it. And then we have from Steve Goff via Julie Pfeiffer, a list of female virologists, which is a Google Doc spreadsheet. I think Julie set this up originally a long time ago. I told her to set up a Google Doc, and she did. So here's the link, which is pretty cool. And you can always reach Julie Pfeiffer on Twitter if you think you should be on it. So that's pretty cool.
Yeah.
Now we are giving away a copy of Viruses by Michael Cordingley. He is the author of this book, which was published by Harvard University Press. And he emailed me. He said, I have 2 extra books. Would you like them? So one of them he signed, Dear TWiV listener, TWiV is a jewel in scientific communication. Keep on listening and learning, support evidence-based science, and tell your friends. Regards, Michael Cordingley. This is going to be worth a ton of money one day.
Absolutely.
Yes.
So what do you think we should do for this? Anyone have a creative idea for a contest? Or should we do the usual X number of emails?
Don't do like 20th email. That took forever.
No. If you cut it off for a time limit, though, and then randomize the number of applicants, then you can give it away.
I can do a time, and then we'll randomize it. That's a great idea. So this episode comes out on the 19th of November.
Right.
And how much time should we give it?
Just give them to the end of the
2 episodes.
To the end of the year.
Or you could give them to December 1st, and then the morning of December 1st, you can randomize.
That's Kathy's birthday.
Send us an email, twiv@microbe.tv, and in that email, the subject line should be Viruses. Which is the name of the book.
The kind that make you sick.
And that's in my license plate, too.
How about the subject line should be Viruses book?
Viruses book.
Yeah, that's the deal.
Yeah, that's good, because other people will send.
Because people emailing you about viruses is not very distinctive.
And if you mention our names on TV, you will send you 2 of them for an additional cost.
All right, so twiv@microbe.tv. You have until December 1st, subject line Viruses book, and you can write whatever you want in the email.
Yes. Technicians are standing by.
Yeah, right.
Yeah, till the morning of December 1st. So you have it in time to
Eastern
do
Standard
it for
Time.
December 1st.
Exactly.
All right, TWiV at microbe.tv. You can find TWiV at Apple Podcasts, microbe.tv. And if you would like to support us, please go to microbe.tv/contribute. There are a number of ways that you can do that. Amy Rosenfeld is on Twitter, ABR22. Thank you, Amy.
Thank you.
Did you enjoy?
Yeah, it was fun.
Did you listen to TWiV, right?
I listen every Sunday morning.
Yeah, one
I
Sunday.
get annoyed when
it doesn't drop on time, right?
Yeah, because I'm here at 9. It's my thing from 9 to 12. Where I'm like setting up plaque assays or getting ready to go slice.
Getting ready to go slice.
Oh yeah,
that's a
slice a few brains.
Lovely. Okay, let's not go any further. And then when you're late, it's like total chaos.
Oh
well, good to hear. Well, thank you for joining us.
Thank you for having me.
Great stuff there. Yeah. Really cool. And I'm not biased.
No, not at all.
Dickson Despommier is at thelivingriver.org and parasiteswithoutborders.com. Thank you, Dickson.
It's always a pleasure, as they would say, and I do say that sincerely, and I really enjoyed it.
I hope you feel better.
I feel okay. I just don't sound great.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun. And it was fun to have Amy on. I like it when you have somebody that I've met in person.
I'd forgotten that. That's
good.
No, I coordinated their trip
Yeah,
to the cloisters.
That's right.
Speaking of trips, we're going to be in Austin.
We are.
And we have to figure out something cool to do with that episode,
That's
right? So
you're not going to go to the Alamo?
What's that?
That's going to be a disappointment.
The Alamo's like an hour and a half from here.
It's a disappointment.
That's in San Antonio.
Right.
So
anyway,
It's in San Antonio.
Right.
Rich Condit is Emeritus Professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
Alan Dove is at turbidplaque.com, and he's also on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiV. I also want to thank the sponsors of this episode, the Department of Microbiology at the Icahn School of Medicine at Mount Sinai in New York City, and the Virology and Gene Therapy PhD program at the Mayo Clinic Graduate School of Biomedical Sciences in Rochester, Minnesota. Introductory music is by Ronald Jenkins at ronaldjenkins.com. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.